no? So, take note ulit na yung apat na levels na to, yung primary, secondary, tertiary, and quaternary structure, ito yung mga considerations natin ha, or determinants natin na dapat natin aralin para maging functional ang isang protein. Okay? So first thing, una tayo sa level 1, which is the primary structure. Okay? Now recall, I have mentioned that the key word for primary structure of protein is amino acid sequence. Okay? Now, yung amino acids in a sequence, recall that they are being held together by a bond. And that bond that connects amino acid to amino acid to amino acid together is what we call the peptide bond. Let me ask you, what is this type of bond? What functional group is a peptide bond? This is an, yes, this is an amide bond. Okay? So I repeat, keyword natin, key term natin for primary structure, amino acid sequence connected together by a peptide bond. Okay, kaya nakalagay dito, defined by the sequence of amino acids. So for example, kindly refer to the figure below. For instance, ito, di ba, 3D man siya nga representation. 3D, three-dimensional representation ng isang amino acids. So we can see here, sa left side muna. Yung nasa left side, di ba, we have here, this is actually the N-terminal. Okay, or let's say the N-terminus. A-terminus. Okay, so yung N-terminus ulit, that is the one with free amino group. Okay, kaya may nakalagay dyan. Imagine niyo lang na yung kulay blue sa buong peptide chain na yan, yung mga kulay blue, that is the amino part. Okay, the NH part. Okay, so ulit, yung N-terminal, yan yung may free amino group. So, etong part na to, okay, in general, yung part na yan ay separated is the N-terminal amino acid. N-terminal amino acid because it is the one with a free amino group. Okay? Non-reacting amino group. And then on the right, let's go to the right part. You have there the, ito yung amino ha. Okay? Free amino group. Free amino group. Itong part naman, ito naman yung free carboxyl group. Okay, the non-reacting carboxyl group. So, free siya. Okay? And ang tawag natin dito ulit is C-terminus. Okay, C-terminus. Okay? And then, may iha-highlight ako na kulay yellow. Itong part na to. Okay, yung highlighted with kulay yellow. Yung part naman yan, that portion that I highlight, okay, I highlighted, that is the peptide bond. Okay, that is the peptide bond. Okay. Another peptide bond, you have here, ito. Ito yung another peptide bond. Ito yung second. And you have here the third. Okay. So, take note, sa isang primary structure, as you can see, you have their amino acid sequence. Diba? You have amino acid sequence, they are being linked together by amide bond, by a peptide bond. Okay, yung na-highlight ko na kulay yellow, mga peptide bond yan. Okay, now focusing on those peptide bonds that I highlighted, it actually has characteristics. Meron siyang features that we have to know. We have to consider. Okay? Now, talking about the peptide bond, okay. Ayan. So, itong mga highlighted na peptide bond, okay? Take note, these are said to be rigid. Okay, rigid. Yan yung characteristic ng peptide bond. Aside from that, it is planar. Okay, it is planar, it is rigid. No? Kapag sinabing rigid, planar, this tells us that ang peptide bond is not capable of rotation. Okay, sulat nyo, not capable of rotation. Alright? Kumbaga, hindi siya basta-basta na rotate yung peptide bond, yung amide bond. Okay? Because again, peptide bonds, they are characterized as being rigid. Okay, rigid sila, planar. Okay. Para mas ma-visualize ninyo ang peptide bond which is not capable of rotation, let's have this slide, yung next slide. Ayan. Okay. So on this figure, okay, let me encircle first the peptide bond again. Okay, the peptide bond. The peptide bond is this one. Ako encircle with kulay purple. Okay, yan yung peptide bond dito sa 3D representation or 3D figure. Again, that is the peptide bond and amide bond. Okay, now recall, okay, yung peptide bond, amide bond, sa previous natin na discussion, ito yung bond na naglilink, nagko-connect ng amino acid to amino acid. Ano yung reacting group ng isang amino acid to another amino acid para mabuo yung peptide bond, di ba? That amide bond, siya yung naglilink particularly ng carbonyl carbon ng first amino acid. Okay, itong carbonyl carbon. Okay, yung carbon dyan. Okay, and then kinoconnect yan via peptide bond to the amino nitrogen. Okay, ng isa pang amino acid or ano yung term natin ng amino acid, residue. Susulat nyo residue. So residue na yan kasi parte na yan ng isang peptide chain. Kapag nag-connect na yan yung mga amino acid, technically hindi na siya dapat tawagin na amino acid. Okay? Kasi nga nag-combine na, okay? So parte na lang siya, residue, moiety na lang siya. Okay? Ng isang large nga or complex molecule of a peptide. So I repeat ha, yung peptide bond ulit, okay, para ma-reinforce sa utak nyo. Itong peptide bond, okay, it is the one that links carbonyl carbon, okay, yung may star, at saka yung amino nitrogen, okay, ng isa pang amino acid residue. Okay, so in this figure, nasaan ba dito yung carbonyl carbon? Okay, or yung mismong carbonyl. Okay, I'll highlight it with color black. Ito yung carbonyl. Okay? So, kumbaga, itong kinulayan ko na black, ito yung carbon ng carbonyl. Carbonyl ha is carbon double bond. Oh, that is the carbonyl. Pag sinabing carbonyl carbon, oh, ito yun. Okay? And then, nasaan ba yung amide NH and amide NH? Ito, asan ba yung amide NH sa 3D structure? Okay? Yung amide NH, ito naman. Okay? So, yan probably in this 3D representation. Again, si peptide bond, yan yung nakakulay violet or unsa man na pink ba na siya sa inyong pananaw. And again, sabi ko kanina, characteristic niya, rigid siya. It is not capable of rotation. Okay? So, any polypeptide chain, okay? Any peptide chain po, okay? Or peptide molecules. Ang peptide bond dyan, amide bond, hindi umiikot ha. Hindi po siya umiikot. Okay? Ibig sabihin, yung mga parts na malapit kay peptide bond, okay, yung hino-hold ni peptide bond na group of atoms, hindi yan umiikot in general. Okay? However, yung buong peptide chain may possibility umikot because of other bonds, not because of a peptide bond. Okay? So, ibig sabihin, kung si peptide bond hindi siya rotational, kasi nga rigid siya in character, meron naman tayong mga rotational bonds in a peptide chain. Okay, I repeat, meron tayong ibang bonds na capable of rotation. Okay? So, umiikot, kumbaga. In fact, there are two. Merong dalawang rotational bonds here. Okay? And what are these bonds? Okay? Una, number one, okay, again ha, rotational. We are now on the rotational bonds. Number one is yung we have the bonds that surrounds the alpha carbon. Okay? Ano ba yung bonds na nagso-surround sa alpha carbon? You know already alpha carbon, di ba? The central carbon of an amino acid. Okay, kung ito yung alpha carbon, ako sang i-zoom out. Ito yung alpha carbon, di ba naa siya 4 groups. Meron siyang default, ayan, carboxyl group, meron siyang amino group, diba? That is the structure of amino acid. Yung rotational bond dyan, okay, is, ng ano ha, sa peptide chain, is ito. So, ibig sabihin, yung mga bonds that surrounds alpha carbon. In particular, number one, we have the bond that connects alpha, ay sa ha, kaya ito sang itarong atong slide. Okay. Okay. So, ulit, number one, we have the bond that connects alpha carbon with amino group. Okay, with amino group. That is a rotational bond. Yung bond ha na nagko-connect sa alpha carbon towards the amino group, that is a rotational bond. Number two is yung alpha carbon, okay, bonded to carboxyl. Okay, yung carboxyl COOH. I repeat, the rotational bonds that we can see in the structure of a peptide chain, okay, are the ones found between the alpha carbon and amino group and between alpha carbon and the carboxyl group. Okay. Now, I want you to focus again. Balik ta sa figure. Okay. Now, you might be confused how to recognize here nasaan yung mga rotational bonds. Kasi nga 3D. Mahirap mang yun. Okay? So, ato lang is, dapat masanay pod mo nga mag-analyze og 3D nga structures. Okay? Don't worry, hindi po lalabas yung 3D ha sa mga quizzes tsaka exam. Okay? So dito, I know you are somehow confused to find here the rotational bonds. Given ulit nga 3D siya. Okay? So para madali sa inyo mag-identify ng rotational bonds for 3D structure, unang step dyan is you need to find the alpha carbon. Okay? Para, kasi yung rotational bonds, di ba? Yun yung nagso-surround sa alpha carbon. Ngayon, kahit alpha carbon, ang hirap, di ba? Ang hirap hanapin sa 3D structure. So how do we know where is the alpha carbon here? So the easier way naman to find the alpha carbon is to find the variable chain, okay, ng alpha carbon. Okay, yung variable chain niya kasi doon lang nagkakaiba, 'di ba, yung mga amino acid. So as you can see here, yung walang kaparehas na structure dito, okay, is yung nasa baba. Okay? Then the rest, na may parehas color nga mga atoms dayon. Diba? So probably na kuha na siya nga part carboxyl kanang na mga pareha, carboxyl na siya nga part, then amino group nga part. Okay? So yung nasa baba actually, that is CH3. Okay? CH3 yan. Ito yung carbon, then hydrogen yan, hydrogen. So CH3. So kumbaga R group yan ng isang amino acid. Yung tanong ko, saan ba naka-bond ang R group ng amino acid? Saan naka-bond yung R group ng amino acid? Sa, very good, sa alpha carbon, sa central carbon. Okay, so ibig sabihin, okay, this would be the alpha carbon. Itong kukulayan ko na, yung kulay gray dyan, yung dark, ano ba, dark, ako na lang kulayan, itong black. Yan na yung alpha carbon. Okay? Ayan. Okay? So, ang tanong, if that is the alpha carbon, I repeat, if this is the alpha carbon, where are the rotational bonds? Nasaan yung mga rotational bonds? Siyempre, those that surrounds the alpha carbon. Okay? So, ulit, if mauni siya ang alpha carbon, then this would be the amino group. Okay? Ito yung amino group. Therefore, ito yung isang, lahi na lang ang color, red na lang. Ito yung isang rotational bond. Yung gi-highlight na ko nga red. Okay? And if that part is a rotational bond in between alpha carbon and amino group, then this part naman is a rotational bond between alpha carbon and ayan, carboxyl. So ito yung carboxyl group. Okay? So that is another nga rotational bond. So yan yung mga rotational bonds given this 3D structure. Okay? So please take note ha, yung rotational bonds that surrounds the alpha carbon. So hunaon lang dahil ninyo ang structure ni amino acid. Diba? What centers the amino acid is the alpha carbon. So ano yung mga default niya na bond towards amino and towards amino and towards carboxyl. Okay, so yan yung rotational. O ngayon, ang tanong, ano yung implication ng pag-rotate? Okay? Ano yung implication ng pag-rotate? Okay, due to these rotational bonds. Okay, take note class. If etong dalawang to, kani nga mga bonds, okay, if these bonds rotate, ang tendency, magro-rotate din yung buong peptide chain. Okay, yung buong peptide chain, magro-rotate yan. Okay? Iikot. Okay? Kung sa question, wala mo makita nga rotate nga word, then yung folding. Okay? Folding just is the same. Okay? Magfo-fold, magro-rotate ngayon yung peptide chain. And then ang chances, or ang tendency nga mahitabo kapag nag-fold ngayon yung buong peptide chain, chances are magkakaroon ng close proximity ang mga atoms. Okay? Magkakaroon ng close proximity yung mga atoms upon rotation. In a chain ha, in a chain. Pag ikot, pag fold ana, magkadugol. Na yung mga atoms nga magkadugol ana. Okay? So close proximity, magkadugol. Okay? So, kung magkadugol, magkakalapit sila ng distance, probably na attraction nga mahitabo from atoms to atoms. Diba? Kay duol naman sila. Okay? And take note na naatay mga certain bonds, certain force of attraction that depends on distance. Kung once magkaduol ga ini sila nga atom, naagi possibility ma-attract sila sa each other. Okay? So isa na doon na force of attraction na kadepende sa distance kung magkadugol na sila is the hydrogen bond. Okay? Kaning hydrogen bond probably dako ang chance nga kung motuyok ang usa ka peptide chain, mo fold ang usa ka peptide chain, hydrogen bond will be formed. Okay? So take note ha, kapag nagkaroon ng rotation, it will cause eventually formation of hydrogen bonds. Okay? Now, ano ba yung hydrogen bond? Okay? Is this related to another level ng protein structure? Na ba siya relationship sa usa ka structure, a protein structure, level ng protein structure? Anyone? Related ba siya sa primary, secondary, tertiary, quaternary? Natulog na may ma'am. Saan sa man? Asa siya related nga level of protein structure? Yung hydrogen bonding, Are you sure? Is it primary? Di ba sabi ko, ang key term natin sa primary is only amino acid sequence, peptide chain. Di ba? Yan yung highlight natin sa primary structure. Very good. Okay? Yung hydrogen bond, yan yung key term natin for secondary structure ng protein. Level 2 na yan ng structure ng protein. So, ang point dito na slide is, tumataas ang level ng amino acid kasi umiikot ang peptide chain due to rotational bonds. Okay? Without rotation of the peptide bond, walang level up. Walang upgrading sa ilahang structure. So, importante din yung folding, yung pag-rotate ng peptide, not peptide bond, ng pag-rotate ng rotational bonds. Okay? Para mo level up, okay, into secondary level. For example, okay, so later, madadiscuss natin yung secondary structure ng protein in detail. Okay? So, since na-introduce ko ngayon yung hydrogen bond in relation to secondary, okay, structure, okay, sige, dito tayo sa next slide. How are secondary structure are being formed? Okay? Number 1, yun na nga, meron tayong mga bonds, 2 bonds, for instance, with reasonably free rotation. Okay, they freely rotate. Yun na nga, mga rotational bonds. Okay, sige, tell me, ano nga ulit yung mga rotational bonds? Alpha carbon towards, and alpha carbon towards, sige, very good, towards amino group, and towards carboxyl group. Dahil sa etong bond na to, dahil sa rotational sila, dako ang chance na secondary structure will be formed. Okay? Now, another thing about this rotational bonds, take note, since these bonds, they do rotate, okay, there is a rotation of angles. Okay? Magro-rotate ngayon yung mga original angles nila. Okay? Now, ang tawag natin sa mga angles na na-produce, okay, Kaya 'di ba kapag na ay rotation, na ay bonding, na magud yung mga significant na mga angles, 'di ba? Value angles nga ma-produce. Okay? Because again of rotation. Now, ang tawag ulit sa angles na yan na nabubuo, angles. Okay? Ang tawag is Ramachandran angles. Okay? Ramachandran angles. Okay? Ayan. Or angles, parang pwedeng ganito yung symbol. Okay? I repeat, dahil nga sa rotation, okay, ng mga bonds, may nabubuong angles. Ang tawag doon sa angles na nabubuo, Ramachandran angle. Now, meron tayong dalawang types of Ramachandran angles. Depende yan kung ano yung nakalink sa alpha carbon. Okay? So with that being said, let's refer on the next slide. So on this figure, okay, let us locate first the alpha carbon. Labeled naman. The alpha carbon is already labeled in the structure. Okay, so kung labeled ng alpha carbon, kabalo na mo, asa ang rotational bond ana. No? So, if that is the alpha carbon, then the rotational bonds are the following. So, that would be between alpha carbon and, ayan, yung highlighted, that is towards the carboxyl group. Okay, so ito yung carboxyl. Naka 3D lang yan, pero carboxyl yan. And then, we have this one, another rotational bond. Okay, yan yung, ito yung amino group. Okay, so again, these are rotational bonds. 1, 2. Okay? So that's why arrow. Makita nga arrow, di ba, sa figure. Yung arrow na yan na nakikita nyo, that represents that bonds are rotational. Okay, that bonds are rotational. And yun na nga, because of the rotation, Ramachandran angles are being formed. Okay, and there are two types of Ramachandran angle. Number one, ang number one na type of Ramachandran angle is this one. Okay, kaning encircled with color green. Kanang murag hugis tinidor ni Satanas. Kita na mo, wala pa ko kita ni Satanas ha, pero kita na ko mga salida, diba? Nga na ang tinidor ni Satanas. Okay, so ang tawag dyan na angle is psi. Okay, psi angle. Okay, so that's one type of Ramachandran angle. Okay, so ang psi angle, everyone, ang psi angle, as you can see, napoproduce siya upon rotation of the bond between alpha carbon and carboxyl. Diba? As labeled. So kapag nag-rotate itong bond na to in between alpha carbon towards carboxyl, may mabubuo na angle. Ang tawag sa angle na yan is psi angle. Okay, psi angle. Whereas, yung nasa baba, I will encircle it with color blue. Ayan. Yan naman, tawag dyan is phi angle. Phi angle. So that's the second type of Ramachandran angle. Okay. Now, regarding naman phi angle, apparently, that is the angle produced upon rotation naman of the bond between alpha carbon and amino group. Okay, so itong bond na to, kapag nag-rotate yan, makakabuo ka ng phi angle. Okay, so I repeat, psi angle and phi angle are Ramachandran angles. Okay, so overall that ends the primary structure. So, tapos na tayo sa primary structure. Okay, so before tayo proceed sa next slide, recall sa ta para paspas ang reabsorption. Okay? So, sa primary structure ulit, sa primary structure, ang atong key term is amino acid sequence. Okay, and they are being held together, amino acid to amino acid, held together by a peptide bond. O, tanong, ang peptides ba are capable of rotation? Ang peptides, okay, very good. No, because they are rigid. Okay, peptide bonds are rigid. Okay, so we only have other, other nga mga rotational bonds that causes folding, that causes rotation of the peptide chain. Okay, so yun na nga, because of those bonds na nagro-rotate, we form angles. Okay, we form angles. Okay, so meron tayong phi at saka psi angles. Okay, question, ano yung kapag na-form ang phi angles, okay, ano yung reason? Rotation of alpha carbon with, okay, rotation ng bond between alpha carbon with, very good, with amino group. Okay, kasi phi angle. Okay? O ano naman tawag sa Ramachandran angle kapag nag-rotate yung bond between alpha carbon and carboxyl? The bond between alpha carbon and carboxyl group. Very good. That is psi angle. Okay? Sige, so next slide tayo. Let me introduce this time secondary structure. Okay? So, under secondary structure, recall, keyword ha. Ayaw ninyo kalimti ang mga keyword. Okay, basing ang inyuhang mga style mag-study kay memorize nyo ang one sentence. Okay? No, keyword lang mo mag-memorize. Okay? So keyword natin for secondary structure is hydrogen bond. Okay, hydrogen bonding. Okay, so take note ha. Always sa secondary structure yun, hindi pwedeng mawala ang principle ng hydrogen bonding. Okay, highlight yun na siya permi sa secondary structure. Bakit nagkaroon ng hydrogen bonds? Related lang yun sa na-discuss kanina. Okay, since may mga rotational na mga bonds, that results to close proximity, okay, of certain atoms, okay, that could have attraction with one another. Okay, so I repeat, dahil nga sa mga rotational bonds natin, that causes close proximity ng mga certain atoms, and yung mga nagkadugol ng mga atoms na ay possibility mag-attract sila forming hydrogen bond. And what are these atoms involved sa hydrogen bond? Okay, so take note, pwedeng mag-induce ng hydrogen bonds are those atoms that are said to be electronegative atom. Okay, I repeat, ang hydrogen bond na pro-produce yan, okay, between a hydrogen and an electronegative atom. So, pinaka-common here na makita nato sa mga amino acid structure, ang pinaka-electronegative dira would be oxygen. Okay, see? Oxygen. And you have learned in your basic chemistry, di ba, nga oxygen is an electronegative atom. Okay? So again, because of these oxygen atoms sa structure ng peptide, okay, or peptide chain, most probably pwede magkaroon ng hydrogen bonds, but only if may close proximity. Only if may close proximity, kung magkakalapit lang yung mga atoms. Okay. Now, how is this hydrogen bond is formed? Okay, specifically, technically. Okay, structurally, yun sa man gyud pag-form sa hydrogen bond. So, ang hydrogen bond, okay, they are formed, okay, between amide, between the amide and H, okay, so amide and H, and then the carbonyl, okay, carbon, okay, of the peptide chain or the peptide backbone. Okay, so halimbawa ulit, 'di ba? I draw here the amide NH. 'Di ba? You have there the amide NH. Okay? So halimbawa, amide NH 'yan ng isang dili siya first, kuan lang isang residue. Okay? So isang residue. You have one residue, yes, of course, any residue, any amino acid, meron siyang amide nitrogen NH. Okay? Amide NH. Okay? And then we have here a carbonyl group. Okay, you have there a carbonyl group. So, let's say, meron to sa isang, ulit, sa isang residue. Okay? O, ngayon, dahil sa folding ng mga rotational bonds, chances magkakaroon ng close proximity etong amide NH sa carbonyl group ng isa pang residue. Okay, supposedly magkalayo unta sila na nilihok man, ni fold man ang peptide chain. O, na tendency layo sila pinakauna, nagdugol na noon sila. Okay, due to folding. Now, kapag nagkalapit sila, okay, specifically, etong hydrogen, okay, hydrogen na siya ha, mura naman na siya kung ano eh. Kadali, hydrogen na siya. Okay, so kapag nagkalapit yung hydrogen ng amide NH, okay, at ang electronegative oxygen, okay, kapag nagkalapit yan, that will form attraction. Okay, that will form attraction. Okay, and ano nga ulit tawag sa force of attraction na yan kapag between hydrogen to oxygen? Ano tawag sa bond na yan? Anong tawag sa attraction? Kapag sinabing attraction, bond yan. Force of attraction, bond yan. That is, very good, hydrogen bond. Okay, so dyan nabubuo yung hydrogen bond. Hydrogen ng isang residue towards another electronegative oxygen ng isa pang residue. Okay, that is why if you check here sa sample drawing, we have here a sample nga illustration sa kani sa inyong right side sa screen. Okay, you can check, you can see here, we have the, ito yung amide NH. Ang amide, in totality, ang amide, ito yung amide. 'Di ba? Amide is a carbonyl, balik-balik na lang gyud tani, no? Carbonyl, then na ay N, or usually na NH. So, pwede na amide na yan. In totality, kanang na broken line nga orange, yan yung amide. Ang atong i-highlight lang is yung NH, part ng amide, NH. Okay, so yan yung amide NH, and then, yung sa kabila, yung nasa baba, di ba, you have there the carbonyl. Ito yung carbonyl. Carbon, double bond, O. Okay? And as you can see, ibang amino acid yan, di ba? Magkaiba sila, magkaibang residue. Residue, another residue. Okay? So, ulit, because of close proximity, okay, nagkalapit yung atoms, may hydrogen bonding na buo. Okay? May hydrogen bonding na mabubuo. Okay? Similar for this side, di ba? This is the carbonyl, oxygen, the amide NH, o tingnan, meron ulit, okay, hydrogen bond. Okay? So, yan yung reason, okay, bakit nagkakaroon ng um, attraction, okay, forming hydrogen bond due to close proximity. Okay? Now, sa secondary structure or secondary level, we actually have two major types of secondary structure. Okay, natay duha ka major types of secondary structure. Ulit ha, secondary paghihapon ta, naalang siya two types and ato ni siyang i-discuss individually. Okay? Question sa, ano nga ulit yung key term natin for secondary structure? Key term nga permi gyud na siya very good hydrogen bond. Ayaw ginakalimte. Okay? So, pag topic nato aning mga major niya nga types, okay, naagihapon ang hydrogen bond kay types nga sila ngang secondary structure. So, these are the alpha helix, okay, alpha helix or alpha helices. When you study, palihog na lang kuha, inyo siyang iana alpha. Alpha is supposedly ing ana. Nya wala man kuy ambot ang akong PowerPoint kay ang pag-insert nako sa symbol nga alpha. Box ang mugawas, ing ana ang mugawas. Dili kuan bitaw, di niya basta. So kabalo na mog sabot. So ang beta also letter B ra akong gibutang. So kamo na lang ha magkuan ana ang beta. Okay, mo ni ang alpha. Okay, so we have the alpha helix and the beta pleated sheets. Okay, alpha helix or alpha helices. Okay, beta sheet, or this is also known as beta pleated. Pleated. Okay, pleated sheets. Okay. Sige, overview lang sa. 9 minutes na lang, no? Sige. I will just discuss until overview. Okay? So yung detail kung manggid kayo ni ang alpha helix and beta bugat siya, medyo bugat siya until tertiary. So overview until diri na lang sa. Okay ra ba? Nga diri ra tama ang atong discussion. Padara po atong padayunon. Okay ra daw. Ganahan pud ko ana. Sige. Ganahan ko sa inyo. Sige, ganahan ko. Sige. So take note, in alpha helix, sa alpha helix, overview lang ha, spiral siya in shape. Okay? Dili na sila pariha og structure ha. Di man gyud maklaro akong kwan. Spiral yun ni actually ang alpha helix. Ang spiral di ba kay mura siya kung ako siyang ipa-vertical, mura siya ana ba? Okay? Spiral. Nakakuan lang na siya horizontal lang na pagka-spiral ka. Basing inyuhang hunaunan pariha ra sila structure sa ubos. No, spiral lang first. Spiral ha, spiral. Spiral in shape. Okay, spiral. Okay. So, next meeting, ako siyang ma-discuss. Ma-visualize ninyo, mas ma-visualize ninyo iyahang structure. And then, often, ang alpha helix, this is composed of only single polypeptide chain. Susulat nyo, single, ano siya, single, only single polypeptide. Okay, polypeptide na siya, polypeptide chain. Okay, polypeptide chain, single lang. Again, composed of only single polypeptide chain. Okay, and then, for beta pleated sheet naman, yung shape niya, zigzag. Kana inyuhang kakakita sa screen, di ba? Zigzag na siya. Okay, so yung beta pleated sheet, zigzag. Okay, yung shape niya. Zigzag yung shape niya. And often, when it comes to composition niya, it consists of either pwede isang polypeptide or more polypeptides yung makikita. Okay? Pero mas common sa beta pleated sheet yung more polypeptides. Okay? So again, it consists of one or more polypeptide. Okay? So I repeat, this is just an overview of the major types of the secondary structure ng protein. Okay, so take note, ang shape po ng alpha helix is spiral. So as mentioned, spiral siya. And when we say spiral, okay, ang kanyang axis of rotation, take note, yung axis of rotation niya is actually vertical. Okay? Axis of rotation of alpha helices, vertical. So, halimbawa, yan, yung in-illustrate ko, di ba, vertical line man yan. So, vertical axis yung axis of rotation, kung paano nag-rotate yung polypeptide with an alpha helix structure. Now, ang alpha helix po, yan ay nabubuo because the peptide chain forms a spiral shape structure. Okay? I repeat, ang alpha helix, nabubuo siya because yung mismong polypeptide chain, it forms a spiral shape. Okay? Sa mismong axis of rotation. Okay? So, kumbaga, ganito siya. Let me illustrate it to you. Okay, let's have this. So, parang ganito siya. Ana. Okay, so yan kibali yung polypeptide. Yung polypeptide, when it forms a spiral shape structure, then that is a alpha helix 1. I repeat ang axis of rotation, yan yung vertical line sa gitna, axis of rotation. And then yung parang loop na kulay white dyan, yan yung alpha helix. So yan basically yung hitsura ng alpha helix. Now, bakit nagkakaroon ng spiral shape? Okay, that is actually because of the hydrogen bond. I repeat, that is because of the hydrogen bonding. Okay? Bakit ulit nagkakaroon ng hydrogen bond? Isang klaseng force of attraction. Diba, remember, kapag nagkakaroon ng close proximity of atoms. Diba? Related lang to sa na-discuss last meeting, no? Pag may close proximity of atoms, then there would be H bonding. Okay? And saan ba nabubuo yung hydrogen bond? Close proximity of electronegative atom na oxygen bonded to hydrogen. Diba? That is a hydrogen bond. Okay? What connects oxygen there and hydrogen ng isang residue, amino acid residue, that's hydrogen bond. Okay, now ngayon, ang alpha helix, isang type ng secondary structure, meron siyang certain characteristics. I repeat, meron siyang certain features distinct nga mga descriptions, okay, about alpha helix. And you have to know this, okay? Characteristic kasi siya ng alpha helix. So, number 1, unang characteristic niya is regarding sa R group niya. Okay, with regards to its R group. Okay, so take note, ang R group or ang variable side chain, tandaan, makikita po sila sa exterior portion ng alpha helix. Okay, so yung mga kulay red dyan, halimbawa, dito natin nakikita yung mga R group. Okay, R group. Okay, so I repeat, yung R group nasa exterior portion of the alpha helix. Okay, nakalabas yung mga R groups, naka-protrude, kumbaga. Okay, naka-protrude. Now with that, how do we relate now the alignment ng R group to the axis of rotation? Paano yung alignment nila? Okay, so tandaan, ang R group po, or the side chain, as you can see on the illustration, yung R group to the in the alpha helix, perpendicular siya. Perpendicular siya to the axis of rotation. I repeat, perpendicular, okay, yung R group to the axis of rotation. O, tanong ulit, nasaan yung axis of rotation? Ano yung kulay sa illustration? Axis of rotation, yan yung kulay, tama, kulay blue sa gitna. Okay? Si R group daw po, tandaan, sa isang alpha helix, ang R group, yung alignment niya, okay, to the axis of rotation, perpendicular. Okay? So please take note, perpendicular sila with each other. R group to the axis of rotation. Okay? Let me break down the terms perpendicular to the axis para mas klaro pa gid sa tanan. Okay, number one, let's tackle on the axis of rotation. Okay? So itong axis of rotation, nasa gitna, di ba vertical line siya, vertical line. Okay? So, yung axis, yung mismong axis, halimbawa, okay, which is in a straight vertical line, pag sinabing perpendicular, so obvious kaayo nga, it will form a 90-degree angles. Okay? Halimbawa, this is the rotation, axis of rotation, and then you have there the R group na nasa exterior, di ba? Yung alignment nila perpendicular. Then we can observe formation of a 90-degree angles. Okay? And I think you are already familiar with kapag sinabing perpendicular, it will really form, di ba? 90 degrees angle. I repeat, perpendicular, it will form a 90 degrees angle. So, pwede nyong highlight dyan. Parang ganito. Okay? 'Di ba? Kung siya, ma-explain siya by just a schematic diagram ng mismong alpha helix. 'Di ba? Makakabuo ka dyan ng somehow 90 degrees nga angle. Okay? Axis of rotation to the R group, perpendicular 90 degrees. Next, another characteristics of alpha helix. Okay, number 2 na ta. Number 2, kulay white na lang. In terms of H bond naman, in terms of hydrogen bonds. Okay? So, syempre, may makakreate na hydrogen bonds kasi isa diba itong halimbawa ng secondary structure. And that is the key point kapag secondary structure. Again, with regards to the hydrogen bond being created, please take note that hydrogen bond is being formed because of the amide NH. Naalala po to. Ang hydrogen bond na bubuo, it is because of the amide NH and also the carbonyl oxygen ng isa pang amino acid residue. So, kumbaga dito nabubuo yung hydrogen bond. Now, with regards to the hydrogen bond, we have to know here the interval. Okay, kailan nabubuo yung hydrogen bond sa alpha helix. Okay? Sa mismong polypeptide chain na naka-alpha helix yung structure, kailan nabubuo yung hydrogen bond. So take note, ang hydrogen bond po, itong hydrogen bond, nake-create siya 4 residues away. Okay, 4 residues away. Okay, 4 residues away mula sa starting point na amino acid. So halimbawa, parang ganito siya. You have a starting point na, ayan, amino acid. Ito yung starting point. Okay? Then, you have this, the first amino acid. Okay? The following ha. Doon ka magbibilang. okay, ng unang amino acid na interval bago mabuo yung H bond. So you have pangalawa, pangatlong amino acid residue, pang-apat na amino acid residue. So doon sa pang-apat na yan, okay, pang-apat na yan, dyan mabubuo yung hydrogen bond. Okay? So halimbawa, sa unang amino acid, meron siyang free carbonyl oxygen. Okay? So yung oxygen niya, magpo-form siya ng hydrogen bond to the amide NH, to the amide NH ng pang-apat na residue. Okay, so ako siyang i-edit ha. So you have, for example, amide NH. Ayan. So dyan mabubuo yung hydrogen bond. Okay? From the starting point and apat na residues away. Apat na residues away. Okay, so later napatay mas 3D, three-dimensional nga representation para mas ma-illustrate yung interval with regards to the formation of hydrogen bond. Okay, next. Another characteristics na kailangan yung malaman with regards to alpha helix is yung turning. Okay, so number 3, dire na lang. Turning. So sulat nyo, turning. Okay? So, ito naman yung number of turns. Okay? Yung number of turns, yung number ng liko. 'Di ba sa alpha helix na yung mga liko? May mga liko. Okay? So, ang pangutana, ilang beses, okay, liliko yung spiral shape. So, it has something to do with the turning. Yan yung turning. Okay? Ilan bang amino acid residue ang bumubuo sa mismong turns? Parang ganun. Okay, so tandaan, nagkakaroon ng turning of alpha helix every 3.6 residues naman. Every 3.6 residues. May I ask you, ano yung residues na sinasabi natin dito? Ano na yung nire-represent ng residues? It is the, very good, it is the amino acid ha. Amino acid. Dapat klaro tanan sa mga terms. Okay? So, I repeat, sa turning, magkakaroon po ng turning sa alpha helix every 3.6. Okay? 3.6 residues. Okay? So, turns will be formed. Okay? So, nakuha. Sige, para ma-check siya, if you do really understand yung 3.6 residues na bumubuo ng turn ng isang alpha helix, halimbawa, okay, magbibigay ako ng example scenario. For example, with regards to turning, merong isang polypeptide chain. Okay, meron kang isang polypeptide chain comprising of 36 residues. Comprising of 6 residues. Yung tanong, ilan, if this is the case, ilang turns do you think mafo-form sa mismong alpha helix? Kapag 36 residues ang bumubuo ng isang polypeptide chain, tama. Okay? 10. You simply divide it to 3.6. Kasi nga, yung turn, nagkakaroon, itong turn na to ba? Itong turn highlighted with violet, okay, nabubuo daw to every 3.6 residues. Okay, 3.6 residues. Kung 36 residues ang bumubuo ng isang polypeptide chain, then ibig sabihin, yung parang representation natin sa turns is meron kang isa, dalawa, 3, 4, 5, 6, 7, 8, 9, 10 turns. Parang ganun. Okay? So, 10 turns. Okay? So, klaro na yung mga characteristics. Okay? So, try naman natin ngayon i-apply all these characteristics of alpha helix that is mentioned. So, balik tayo sa figure. Sa unang figure. Ayan. So, in this figure, as you can see, this tells us actually focusing more on hydrogen bond. Okay? I repeat, itong figure nito, this tells more about hydrogen bonding. So, as mentioned kanina, ilan nga ulit yung residues interval bago mabuo yung hydrogen bonds? Hydrogen bonds. Okay, tama. 4 na residues. Okay, so i-verify nato. Tanaw nato ning 3D nga representation kung 4 ba talaga na residues away. Tapos may hydrogen bond na. Okay? So, first thing we have to look for, of course, the electronegative oxygen atom. Okay? For example, ito yung alpha carbon. Yung mga blank dyan, yung mga bilog na blanco, okay, that is alpha carbon. So, i-label nyo dyan, that is alpha carbon. Okay, alpha carbon. So, kung ilan yung alpha carbon dyan, okay, yan yung bilang ng mismong residues in a polypeptide. Okay, kasi di ba each amino acid has a central carbon, has an alpha carbon. Okay, and from this alpha carbon, so may di naman maklaro akong kadali lang ha. Ayan, so this is alpha carbon, alpha carbon. Okay, and we all know na yung alpha carbon, it is being surrounded by R group. Okay, so you have here the R group. Check, meron. Meron siyang N-terminus, the non-reacting, no? Non-reacting N-terminal. Okay, and then meron siyang carboxyl group. Okay, so ito. Yan yung carboxyl group. So you can see on that carboxyl part, okay, meron siyang carbonyl oxygen. Okay, so itong part na to, yung kulay pula, yung nasa likod, di ba may oxygen. Yung mga kulay pula, oxygen yan ha. Yan yung electronegative atom. Okay, electronegative atom oxygen. And take note, di ba, nabubuo yung hydrogen bonding sa mismong electronegative atom oxygen towards hydrogen. Okay? Towards hydrogen of another residue. Okay? So, yan yung starting point, kumbaga. Starting point amino acid. So, I repeat, etong hinighlight ko, yan yung carbonyl oxygen ha. It carries the carbonyl oxygen and it has a tendency ha na to form a hydrogen bond. Saan? Sa mismong amide NH. Okay? So, ito yung magiging source, kumbaga, ng hydrogen. O, saan to makikita? Saan to makikita kung ito yung starting point natin sa pang-4, no? Pang-4 nga residue. Kasi nga, nabubuo itong bond na to, itong hydrogen bond na to, 4 residues away. Okay? 4 residues away. So, let's verify now if 4 residues apart yung formation ng hydrogen bond. So, let us count. Halimbawa, ayan. So, mula sa, of course, this is the starting point. Okay? So, hanapin natin yung central lang ha, central carbon. Okay? The central carbon, the alpha carbon of each amino acid residue. Okay? So, ito yung dulo. Then, ito yung, so, start counting. Ito yung unang interval, okay? Unang amino acid residue. Pangalawang amino acid residue. Okay? Pangatlong amino acid residue and we have here pang-4 na amino acid residue. O, ibang kulay na lang yung starting point. Ayan. Okay? Sige. So, from here, as you can see, focusing on this 4th. Okay? This is the 4th. Okay? Residue. Focusing, makikita natin na meron siyang side chain, tama? Meron siyang R group, yes. Okay. And then, meron siyang NH. Diba? Ito yung NH niya. Okay. Yan yung NH. Amide NH. And I have mentioned Yung pang-apat na residue na yan, siya yung magiging source ng amide NH, yung amide hydrogen to be particular. Okay? So, dito mabubuo ngayon yung hydrogen bonding, okay, between this electronegative atom sa starting point, okay, towards hydrogen atom. Di ba? Mayroon kayong makikitang dots. O, kana akong gi-highlight na kulay blue. Ayan. So, yan yung hydrogen bond. So, di ba? 4 residues in between. Tsaka ma-form, ma-create yung hydrogen bond. Okay? Ayan. Okay, so yan yung ibig sabihin ha ng formation of H-bonds. Pagitan, nakapagitan is 4 residues. 4 residues away. Alright? Okay. Now, kung skeletal yung structure being presented, so halimbawa, I'll draw a skeletal one. So you have here the axis of rotation. We have here the axis of rotation. And then, yung kulay green halimbawa, ito yung, ayan, alpha helix. Okay, alpha helix. So, nakakreate po daw yung hydrogen bond between oxygen, okay, and hydrogen. Between oxygen and hydrogen. Parang ganito yung nangyayari. Okay, and that is every 4 residues apart, every 4 residues interval. Okay, so with that being said, we can connect really the alignment of hydrogen bond with that of the axis of rotation. Okay, and that is, this time, is it parallel or perpendicular? Okay, yung H-bond. Ito yung H-bond, yung kulay blue. Okay, yung axis of rotation is kulay pula. Okay, kulay pula. Then, same direction sila ng hydrogen bond. Tama, it is parallel. It is parallel. Okay, o diba, naglibog na mo. Kaya gibalik-balik na kung perpendicular ganina. Okay, di ba yung perpendicular, nabubuo yung perpendicular nga alignment if they connect, okay, with each other. Halimbawa, ito yung axis of rotation. Then you have on the exterior part, okay, the R group. Ito yung perpendicular kasi nakakabuo ka ng 90 degrees angle. Okay, di ba? Nakakabuo ka dito ng, sa R group yun, alignment between R group towards the axis of rotation, perpendicular sila. Okay? But, di ba, hydrogen bonding yung tinatanong ko, and in-illustrate ko naman, di ba? And yung alignment nila, hindi sila nagko-connect. Parallel sila with each other. Okay? Parallel sila with each other. I repeat ha, yung hydrogen bond, kulay blue, dito. Okay? And yung kulay pula, axis of rotation. So really, parallel sila, no? Axis of rotation and hydrogen bonds, parallel. Parallel yung alignment. Parallel yung alignment. Okay? And ano yung sabi ko sa turns? Turns of this spiral shape. Kailan nabubuo ulit? Yung turns, Yung turns, tama. Nabubuo siya every 3.6 residues. Okay? Every 3.6 residues. Okay? Another figure is this one. This actually summarizes what I discussed earlier. So, discuss na to earlier. First, itong nasa gitna, ito yung axis of rotation, kumbaga. Okay, yung highlighted na kulay blue. So, you have there the axis of rotation here. And then, the R group, look at the R group. Labeled naman, di ba, yung mga R group. Nasaan sila? Nasa exterior. Okay, protruding sila outside the alpha helix. Diba? We can see them. Okay, they lie on the exterior portion. Therefore, alignment nila ulit is perpendicular with the axis of rotation, ha? Alignment ng R groups to the axis of rotation, perpendicular. And then, what about the hydrogen bonds? O, tingnan yung hydrogen bonds. If this is the axis of rotation, parehas lang sila, parallel. 'Di ba? Hindi nag-connect yung alignment nila. So that is actually parallel. 'Di ba pag parallel, ganyan or ganyan. Okay? So parallel sila. Yung hydrogen bond, hydrogen bond to the axis of rotation, alignment, parallel. Okay, and then the turns, okay, per turn po, 3.6 residues. Okay, para mabuo yung turn. Okay, so yan po yung alpha helix in general. The descriptions, the characteristics of alpha helix. Okay. Now, for these slides, na-discuss na po ito lahat. Okay, ikuan lang siya in a statement form. Okay, so as stated here, di ba, hydrogen bonding produces helical coiling. It produces helical coiling or helical meaning spiral shape, okay, of the peptide backbone such that the R groups lies on the exterior. The R groups lies on the exterior of the helix and kung saan perpendicular siya to the axis of rotation. So yun lang yun. Na-discuss natin yan, di ba? Okay. And then sabi kanina, from the N-terminus, okay, from the N-terminus, the carbonyl oxygen of each amino acid residue is hydrogen bonded to NH. Okay, it is hydrogen bonded with the NH. Okay, and that is 4 residues away, di ba? 4 residues away. Okay, yung nabubuong hydrogen bonding between the carbonyl oxygen, amide, and H. Excuse me. So, na-elaborate na to lahat. Okay, na-elaborate na siya. So, yan again, in general, yung alpha helix. Okay. Now, let me emphasize that alpha helix po, it is being formed because of different amino acids. Okay. Ang alpha helix, it can be formed with different amino acid residues. Okay. However, it has to be realized that hindi po lahat ng amino acid, hindi lahat ng amino acid can be able to participate in a alpha helical formation. Okay? In fact, meron tayong mga halimbawa ng mga amino acids that could damage. Okay? It could damage, it could destroy alpha helices. Kaya hindi sila favorable to be present sa isang peptide chain forming alpha helix. Okay, I repeat, hindi sila favorable to form alpha helix. So number 1 here, we have proline. So take note. Proline, recall, is it an amino or amino acid? Okay, diba? Proline is an amino acid, to be specific. Okay, parte din siya ng 20 amino acids, standard amino acid. Pero to be specific, structurally speaking, di ba, isa siyang amino acid. Now, why is it proline cannot join, cannot participate hydrogen bonding? Particularly, it cannot participate intrachain. Okay? Si proline po, hindi siya nililimit niya po yung pagbuo ng hydrogen bonding. Kaya hindi siya favorable sa isang intrachain hydrogen bonding. So I repeat, yung proline, okay, it is not able, okay, it cannot participate in intrachain hydrogen bonding. Okay? So with that being said, kung hindi siya nakakapag-participate ng H bonding, mawawala ng sense yung alpha helix. Kasi 'di ba, ang alpha helix isa siyang halimbawa ng secondary structure. Ano yung key point natin kapag secondary structure? Dapat present yung H bonding. Diba? So if absent yung H bonding, hindi niya kaya mag-participate in H bonding, then madedefeat kumbaga yung definition ng alpha helix. Kasi nga, hindi niya kaya ng H bonding, hydrogen bonding. Next, we have the glycine. It is also not favorable to be present as part of alpha helical structure because it makes the alpha helix flexible. It makes the alpha helix flexible. And supposedly, ang isang alpha helix po ay rigid. Ang isang alpha helix ay rigid. Okay? Next, we have lysine, arginine. Lysine and arginine. Question, basic or acidic amino acid? Lysine and arginine. Tama, it is basic amino acid. Okay? And then glutamate and aspartate residues or amino acid. Ito naman yung acidic amino acid. Okay? So recalling, these are actually charged, no? Knowing a basic amino acid sila, acidic amino acid sila, ibig sabihin charged sila, 'di ba? Charged amino acid. Okay? So particularly, ano yung charge ng basic amino acid? Sige nga, ano yung charge ng basic amino acid? If they accept proton, tama. So positively charged. Okay? If they are able to accept proton, okay, proton is positive, no? So they will really become positively charged. And then kapag naman acidic, negatively charged. Okay? Now, ang reason why this charge, no, charge na mga amino acids do not favor, they do not participate alpha helix formation because kapag nagkaroon ng close proximity with one another, okay, halimbawa, lysine with arginine, di ba, parehas silang basic. Okay, so kapag nagkaroon ng close proximity, same charges, do you think mag-a-attract? Yes or no? Kapag same charges, may attraction pa? No. So, there would be repulsion. Okay, repulsion will happen. Kaya nakalagay na dyan, electrostatic repulsion might happen. Magre-repel sila. Especially for those charged group with the same signs. Okay, so hindi nila na-favor ang alpha helix formation. And then finally, you have their valine, isoleucine, and threonine. Okay, so this amino acid naman, they cause steric repulsion, causing disruption, damage of alpha helix. So taken all together, ito ang amino acids that do not participate alpha helical formation. Okay? Now, saan usually nakikita yung mga alpha helix? Usually nakikita sila sa mga biological or complex protein like keratin and hemoglobin. In fact, sa keratin, almost 100% po ng keratin ay alpha helix. And for hemoglobin naman, 80% is made up of a secondary protein structure, particularly alpha helix. Okay, particularly alpha helix. Although, some portion of it, di ba, hindi 100% nakalagay. Some portion of it are not alpha helix. Later on sa, maybe next meeting, no, matatakol na natin yung mga fibrous, globular protein. So, doon natin marirealize ano pa yung other, okay, structures ng hemoglobin. Okay? So, malalaman natin doon ano yung mga nagko-comprise ng remaining 20% ng hemoglobin protein. Okay, so again, these are examples of proteins containing or made up of alpha helix. Okay, they made up alpha helix. So overall, that's the ending, no, of our discussion on alpha helix, Okay. Now, before ta mag-proceed sa second type ng secondary structure, sige, recap lang sa ta. Sige, recap. Sa alpha helix, no? Ano ulit yung shape? Sa alpha helix, ano ulit yung shape? Alpha helix, tama, spiral. Okay, now with regards to the alignment, doon tayo sa alignment ng axis of rotation. Okay, tell me if it is perpendicular or parallel. Tell me ha, if parallel ba or perpendicular. To the axis of rotation ha, to the axis of rotation. Okay, first, wala pa gani. Bakit may vertical? It's either perpendicular or parallel. Okay. So what about the alignment of hydrogen bond? To the axis of rotation ha? Okay, tama. Parallel. Okay. Kapag naman R group,
Parallel.
R group, perpendicular. Tama. Okay. And saan naman nakikita yung mga R groups? Interior, exterior. Interior, exterior. Tama. Sa exterior yung mga R groups. Oh, ano yung related sa 3.6 residues? Ano yung related sa 3.6 residues? Turns or hydrogen bond? Okay, it is turns. Okay, 3.6 residues, turns. O, kapag naman H bond formation, hydrogen bond formation, how many intervals ng amino acid residue? Okay, tama. So, 4 residues away. Bago mabuo, makreate yung hydrogen formation. Okay? Next tayo. Proceed naman tayo sa second major type ng secondary protein structure. I repeat ha, nasa secondary structure pa rin tayo. But this time, yung isang type niya, isang klase ng secondary structure. So we call this the beta-pleated sheet. Okay? So dun muna tayo sa arrangement ng beta-pleated sheet. Number one, bakit pa siya tinawag na pleated? Okay, bakit siya tinawag na pleated? This is actually related to the term pleats. Okay, this is related to the term pleats. Familiar ba ang tanan sa term na pleats? Familiar ba sa word na pleats? Pleats has actually something to do or synonymous siya to the word folds. Okay, pleats, folds. Kung inyuhang nadumduman kanang sa tela, kanang bitong sa mga palda, sa mga sayal nato sa high school, I'm sure daghan kay nag-pleats, 'di ba? Pleated ang ato ang mga sayal sa high school. Mo siya pasabot sa pleats, no, yung folds. Okay, yan yung mga folds sa palda, halimbawa, yan yung pleats. Okay, yan yung ibig sabihin ng pleats, folds. Okay? Now, ano yung relevance nito? Okay, ano yung importance ng term beta-pleated sheet? Okay, recall na ang shape, 'di ba, ng not amino acid, ng beta-pleated sheet, yung shape niya is zigzag. Okay? And then remember, hindi lang siya isa, tama, sa beta-pleated sheet. Sabi ko doon, pwede siyang present na more peptide chains. Hindi siya katulad ng alpha helix. Yung alpha helix, isa lang na polypeptide chain. Kapag beta-pleated sheet, hindi lang isang polypeptide yung present, pwedeng one or more peptide chain. So halimbawa, ito yung polypeptide, halimbawa. Okay? Ayan. So halimbawa, you have a polypeptide na bumubuo ang pangit. Ako, atroho na. Atroho na ako. So polypeptide, lugar ni siya, okay, in a beta-pleated structure. Okay, so you have a first, okay, polypeptide, and then a second polypeptide, and then you have their third, ayan, polypeptide amino acid residue. So as you can see, ang shape niya, di ba, zigzag. Ang shape niya, zigzag. And again ha, one or more polypeptide chains. Okay? Take note, for this schematic diagram, a zigzag shape of a beta-pleated sheet, ang hydrogen bond po pwedeng makreate in between, in between the structure. Okay? So, ayan, halimbawa, ayan. So, posibleng mabuo dito yung mga hydrogen bond in between the structures, in between the polypeptide chains. Okay? Pangit naman. Sorry ha. Dili ko kabalo mag-jojo. Ana na lang. Ayan. So, halimbawa yung mga broken lines dyan ha. Mga hydrogen bond. So, somewhat like that. Okay? Now, as you can observe, kung inyong i-widen, I hope kanang naamoy kanang very imaginative nga mind. Kung inyong lantawon akong gidrawing, I hope, no? Kung inyong lantawon in a 3D way, more siyag folds. Okay, more siyag folds. Sige, ako i-on akong cam. More siyag yung ani, nag-fold ko og papel, and then, ayan. So, tingnan po yung, ayan. So, parang ganito siya. I hope inyo siya nakita. Mao ba? Makita po ninyo sa ayan, mura siya nga ba naka-fold. Oh, 'di ba? Oh, so I hope inyong na-imagine sa drawing, na 'di ba? Parang merong mga folds, mga pleats. Ayan, dahil sa pagbuo ng H bonds. Ulit ha, yung mga dots diyan, mga hydrogen bonding. So I repeat, nabubuo yung because of hydrogen bonding, okay, in between polypeptide structures, nabubuo yung folds, no? Nabubuo yung folds. That's why pleats, no? Beta-pleated sheets. Okay, beta-pleated sheets. Label na ako dire. These are H bonds ha, para di malibog. H bonds yung dots. Ano naman ngayon ang axis of rotation ng beta-pleated sheet? Okay, this time, listen ha, tandaan, ang axis of rotation po ay horizontal. Klaro man nakahiga man, di ba? Ang structure ng secondary structure, nakahiga yung presentation. So, yung kulay violet, so may color ana sa inyo, kulay pink ba na siya? Akong gi-highlight, nga pa horizontal, yan yung axis of rotation ng isang beta-pleated sheet. Okay, yan yung axis of rotation. Take note, horizontal ha, axis of rotation. Relate nato karon ang alignment. If we relate the alignment of the axis of rotation, okay, sa hydrogen bonds, okay, relate nyo yung alignment ng axis of rotation sa hydrogen bond. Kumusta? Is it still parallel or perpendicular already? Yes, very good. Okay, it is already perpendicular. Very good. No? Akala ko kasi yung sagot nyo is parallel pa rin kasi nakahiga na yung structure dito. Diba? So, perpendicular pa rin to. No? Perpendicular. So, again, alignment ng hydrogen bond to the axis of rotation is perpendicular for beta-pleated sheet. Sa alpha helix, ano yung perpendicular? Sa alpha helix, ano yung perpendicular to the axis of rotation? Tama. It is the R group. Very good. Okay, so take note of that. Proceed tayo ngayon sa mga notes. Okay? So sabi dito, beta-pleated sheet, it is composed of 2 or more, di ba? Different regions of stretches. At least 5 to 10 amino acid residues such that yung hydrogen bonds, sabi dito, yung hydrogen bonding residues are present adjacently opposite to the polypeptide backbone, in which yung hydrogen bonds are perpendicular to the peptide backbone. Yung peptide backbone, yan lang po yung polypeptide chain. Okay? Polypeptide chain lang po yan ha, yung peptide backbone. Okay? Ayan. Okay? Sige. Oh, with regards to the polypeptide backbone sa isang beta-pleated sheet, ano yung movement ng chain? Is it horizontal or vertical? Is it pa horizontal or vertical? Yung mismong chain ha, sa beta-pleated sheet, it is, yes, horizontal. Diba pa horizontal yung chain natin dito, yung kulay red. Okay? Ito yung mismong polypeptide chain, naka-zigzag shape kasi nga yung structure niya is beta-pleated. Okay? Oh, another figure, okay, that explains the structure of beta-pleated sheet. We can see here that the polypeptide backbone, okay, and ayan, similar siya sa alpha helix, no? Similar siya sa alpha helix na ang R group ay exterior. Tama? Di ba sa alpha helix, yung R group, nasa exterior din. Yung R group ha, yung R group nasa exterior part ng alpha helix. Ganon din dito sa beta-pleated sheet. Yung R group nasa labas, no? Nagka-protrude sa exterior portion ng beta-pleated sheet. Kaya lang, this time, ang orientation nga lang ng beta-pleated sheet, yung axis of rotation ha, yung axis of rotation, pahiga, naka-horizontal yung beta sheet. Okay? Oh, kung sa alpha helix, 'di ba, ang axis of rotation is vertical. The R groups nasa exterior. Okay? So, ang alignment dyan, perpendicular. Sa alpha helix ha, sa alpha helix. R group towards the axis of rotation. Now, let me ask you, looking at the beta pleated sheet, so, yung mga exterior portion ha, mga R group yan. So, question, ano yung alignment ng R group to the axis of rotation in the beta pleated sheet? Is it parallel or perpendicular? Okay. Mali po. Okay. It is actually perpendicular pa rin. Tingnan ulit yung structure. Nakahiga lang po yung, 'di ba? This is ako i-change ang orientation na, 'di ba? Mauna siya ang polypeptide chain ng beta pleated sheet. Kumbaga yung axis of rotation, itong kulay black. Okay? So, pahiga. Saan yung R groups? R groups are nasa exterior. Tingnan ulit yung structure. Ano yung alignment ng axis? Ano yung alignment ng axis of rotation sa mismong R group? Perpendicular or parallel? Take 2. It is perpendicular pa rin. Okay, perpendicular pa rin po. Okay, perpendicular pa rin. Parehas lang actually sa alpha helix. Kasi nga sa alpha helix, di ba? Naka-vertical lang naka-vertical yung axis. Naka-vertical yung axis. But look at the R groups. Nasa exterior din naman yung mga R groups. Di ba nakakabuo ka rin ng 90 degrees angle. So, di ba kapag may 90 degrees angle, perpendicular yan. No, perpendicular. Tingnan yung sa beta pleated sheet yung nasa right portion. 'Di ba nakakabuo din tayo dito ng 90 degrees angle. 90 degrees angle. So this is still perpendicular. Okay, perpendicular. Again ha, alignment ng axis of rotation to the R group for beta pleated sheet, perpendicular. Okay? Sige nga. Sige, icebreaker sa ta, icebreaker. Q&A sa Q&A. You tell me if it is alpha, if it is beta, or both. Okay, based sa akong discussion ha. Ito lang yung i-type nyo. Alpha, pwede letter A. Okay, letter B for beta, then whole word na both if applicable siya sa both alpha and, beta. Okay? Number 1. Hydrogen bond is perpendicular to the axis. Hydrogen bond is perpendicular to the axis. Tama. It's beta. Yung kanina lang na slide. Okay? Hydrogen bond is parallel to the axis. Hydrogen bond is parallel to the axis. Tama. This time it's alpha. Zigzag shape. Okay, tama. Zigzag shape is beta. Kapag spiral, tama. Spiral is alpha. Helical. Helix, so alpha. Okay, alpha. Okay. What about R groups? R groups lies on the exterior. R groups lies on the exterior. Tama. Both. No? Both. Nasa exterior yung R group sa both alpha and beta helix. R groups is perpendicular to the axis. R groups is perpendicular to the axis. Okay, tama. It is both also. Okay, both. Okay, good. Sige. So, let us now continue. Okay, continue ta. Okay. Now, we have mentioned earlier na ang beta pleated sheet, diba, pwede siyang one or more polypeptide chains. Okay, yan yung beta pleated sheet. Okay. Now, may connection yan sa nabubuong hydrogen bonds. Okay, may connection po yan sa nabubuong hydrogen bonds. Una, kapag yung hydrogen bonds ay interchain, it is expected na more than one polypeptides yung nag-participate. Okay? More than one. Susulat nyo, than one. Okay? From the word inter, di ba sa chemistry, kapag interaction, di ba? That's between, in between, no? In between. So, kumbaga, yung hydrogen bonds that is being formed in between several, no? Separate polypeptides. In between, okay? One polypeptide chain to another polypeptide chain. So that's interchain bonds. Okay? Now, kapag naman intra-chain, it is expected na ang na-form na polypeptide chain sa isang beta pleated sheet ay isa lang. Okay? Isa lang na polypeptide chain. From the word intra. Diba? Pag sinabing intra, within. No? Intra is within that single chain. Okay? Now, between these two interchain bonds, intrachain bonds, mas common po sa beta pleated sheets, yung inter. No? Mas common yung interchain. Okay, that's for beta sheets. Kasi again, for inter, more than one. Okay, na-favor nila yung more than one polypeptide chain. Whereas, common naman sa alpha helix, okay, alpha helices, yung intra chains. Okay? Now, another concept that we need to discuss under beta sheets, that is the arrangement. Paano yung arrangement of those chains? Okay, ng mga polypeptide chains. Under beta pleated pa tayo ha, beta pleated sheets. In particular, kaninga concept, applicable na siya sa multiple chains. No? Multiple chains, polypeptide chains. Multiple, more than one. Okay, I repeat, kani sila nga arrangement, ani nga slide, applicable lang ni siya sa multiple, more than one polypeptide chain. So for instance, for example, we have a multiple peptide chain like you have 1, 2, then pangatlong chain. Okay, so my chain, and then for us to identify its arrangement, if it is parallel ba or anti-parallel, simply know the location of the N-terminus and the C-terminus. So you know this already, di ba? N-terminal, C-terminal, N-terminus, C-terminus. So for instance, sa first given nga illustration, okay, halimbawa yung N-terminus for the first chain is formed here. Nandito yung N-terminus. Okay? Then, dito yung C-terminus. The same with second and third. Dito yung N-terminus, C-terminus, and C-terminus. So, the same sila. Pag ganito yung arrangement, obviously, this is parallel. Okay? Kapag same direction, okay, that is parallel. Parallel beta pleated sheet. Okay? Next, kapag naman opposite, halimbawa, say for instance, I have here a first chain, then the second chain. Sa first chain, halimbawa, you have N-terminus here, then C-terminus. And then for the second chain this time, you have here the C-terminus, then here would be the N-terminus. Kung titingnan natin yung direction, di ba, this time, opposite, no? Opposite direction. So, ang tawag naman dito is anti-parallel. Okay? Anti-parallel na beta pleated sheet. So, it has something to do talaga with the location of the N, location of the C terminus. Kung same direction, parallel. Okay? Kung opposite direction, anti-parallel. Okay? Now take note, regarding this parallel and anti-parallel sheets, they actually have implications. Meron silang implications or effects, kumbaga. Particularly, may effects sila sa hydrogen bond. Halimbawa, for the first figure, ito, next slide. Ayan. For the first figure, letter A. Okay. As you can see, may I ask you, sa first figure, ano ba yung direction ng chain? Same? Different? Parallel? Anti-parallel. So, same. Tama. Diba? Kung ako i-box, ayaw ninyo tanawa ang structure. Okay, tanawa lang tong label, no? Yung direction ng N-terminus, ng chain ba? Okay, kung same ba sila, nagsimula sa C-terminus to N, C to N. So, pareha sila. Okay, labeled naman. C to N, C to N. So, it is parallel. It is parallel. Now, regarding the first figure, ang ratio ng hydrogen bond is actually 2:1. Okay? Ratio ha, ang ratio ng hydrogen bond is 2:1. Klaro man po sa structure, no? May nakabox sa taas na dalawang residue. Sa baba na chain, isang residue. 2:1. Because the first chain, on the first chain, dalawang residues ang nag-share, no? Nag-share sa isang one residue ng second chain. I repeat, yung makikita natin dito sa illustration, sa first chain, okay, dalawang residue ang nag-share, okay, sa one residue ng second chain, forming hydrogen bond. Okay? So kapag parallel direction, parallel sheets, okay, yung iyahang gina-affect sa hydrogen bond formation is 2:1. 2:1. Whereas yung nasa baba naman, obviously this is anti-parallel, no? Anti-parallel kasi opposite direction, di ba? Opposite direction. So letter B naman, kapag anti-parallel direction, take note, 1:1 naman. 1:1 yung hydrogen bond. 1:1. So perfect nga interaction, no? As you can see, perfect interaction, equal interaction, kumbaga, equal interaction. Okay? Ayan. So that is the difference between the two, parallel and anti-parallel. Parallel and anti-parallel. Now, in complex proteins, there are beta sheets that can be found, okay, sa mga complex type of protein. So, example, mga fibrous, okay, mga proteins, and then the globular proteins. We will discuss this, no, thoroughly, mga fibrous globular proteins, pagdating natin sa maybe by next week, no, next week. Halimbawa lang muna ng fibrous, example lang ha ng fibrous proteins, collagen. Collagen. That is a fibrous protein, no? Collagen, elastin, ito yung mga halimbawa. Keratin, yan yung halimbawa ng mga fibrous protein. Okay? Halimbawa naman ng globular proteins is myoglobin, hemoglobin. I repeat ha, nagbigay lang ako ng example, pero hindi ko sinasabi na lahat yan, okay, meron talagang beta sheets. Okay? Ang sabi lang naman dito sa slide, beta sheet can be found, no? Pwede siyang ma-found in fibrous type of protein. So, most probably, pwedeng sa elastin, meron siya. Kasi sa keratin, di ba, sabi natin 100% ng keratin, it's alpha helix, di ba? Alpha helix. So, sa globular proteins, meron tayong makikitang beta sheet, no? Nga structural protein, most probably sa hemoglobin. Ayan. Okay. So, again, ma-discuss natin siya. Natin siya separate segments sa itong discussion next meeting. Another example of protein with pleated sheet structure is yung amyloid protein. So, amyloid protein, this is composed of twisted beta pleated sheet and a 3D arrangement. Okay? Yung 3D arrangement niya is identical to that of the silk fibrils. Okay? So, yan yung mga halimbawa kung saan pwede natin makita yung beta pleated structures. Okay, so altogether, that ends our discussion on alpha helices and beta pleated sheets. Okay, so again, these two alpha and beta pleated sheets, secondary structure po to sila. Secondary structure. Okay, oops. Ayan. Alright. Sige. So, na-discuss na natin yung dalawang klase ng secondary structure. The alpha and then the beta. Again major types to sila ng secondary. Secondary level, secondary conformation ng protein. Okay? Yung alpha 1, alpha, di ba, is alpha helix, spiral. Yung beta pleated sheet, zigzag. Okay, zigzag. Okay? Now, ang tanong, ano yung connection nito, yung alpha helices, beta sheets, sa loops, turns, and bends? Okay? Ano yung connection niya? Okay. Yung loops, turns, and bends po. When we talk about alpha helices and beta pleated sheets, when they fold, okay, when they do folding, Okay, di ba nagfo-fold yung mga polypeptide chains, depende sa structure nila. Yung loops, turns, and bends po, very important sila to facilitate connection. Okay? To facilitate connection. Halimbawa, kung you have here a beta pleated sheet. Okay? Sige, dagdagan natin. Another beta pleated sheet, and then you have here the alpha helix. Okay? So yan, kapag ganyan lang, walang mabubuong connection between these secondary structures. So yung gagawin ng loops, turns, bends, magfa-facilitate sila ng connection, linkages, no, between these secondary structures. So importante po yung loops, turns, and bends, no, para ma-connect sila. Okay, for loops, sabi dito, these are regions that contain residues beyond the minimum number necessary to connect, no, to connect adjacent regions of secondary structure. Yung point lang po dito is yung loops, nagko-connect siya ng alpha helices, okay, and beta pleated sheets. Okay, for connection purposes. Okay, yan yung gawa, no, yung role ng loops. For turns and bends naman, ganun din naman, same concept for connection, okay, ng mga secondary structures, okay. But for this is more common. Yung turns and bends, very common naman siya sa mga adjacent strands of anti-parallel beta sheets. Okay? So, common siya sa adjacent strands of beta sheets. And specific with beta turn. Beta turn, this involves 4 amino residues. Contains 4 amino residues in which, take note, the first amino acid residue is hydrogen bonded. Okay, it is hydrogen bonded to the fourth amino acid residue, resulting to 180 degrees turn. Okay? So, yan yung beta turn. Okay? And with regards to beta turn po, take note, ang amino acids na usually talagang present sa beta turn, often we can see proline. I'm sorry for that background noise. Proline and glycine. They are often present po in beta turns. Okay. Bakit sa beta turns present yung proline tsaka si glycine? With regards to proline, di ba si proline yung characteristic niya, hindi siya nagpa-facilitate masyado ng hydrogen bond. So, ibig sabihin, pwede siya for beta turns kasi nililimit niya lang yung hydrogen bonding. Okay? And that is very important for beta turn. Kasi nga, the hydrogen bonding is nabubuo from first residue to the fourth residue. So, kumbaga nalilimatahan niya yung hydrogen bonding formation. And with glycine naman, ano yung characteristic ng glycine na nasabi kanina? Glycine is flexible, tama? Flexible yung glycine. And that is very important for beta turns. Kaya nga bendable, flexible yung beta turns. It's because of also the presence of glycine amino acid. So, tingnan niyo yung notes yung kanina. Flexible po yung characteristic ng glycine. Okay, so overall, yan yung importance ng loops, turns, and bends. So, yung general idea lang talaga nila is for connections ng mga secondary structures. Importante sila. Kasi the moment na mag-fold yung mga secondary structures, siyempre kailangan ng connection, no? Connection. Paano sila magko-connect? Paano sila magla-level up? 'Di ba? Kung walang bridges, no? Walang bridge. Okay, so yan yung importance nila. Okay, so done with loops, turns, and bends. Next, sige. So with regards to alpha and beta structures, diba na-discuss natin yung mga certain characteristics nila. Na-discuss natin yung features, characteristics ng isang alpha helix. Ano yung basic with regards to alpha helix. Ano din yung mga basic information about beta pleated sheets. Okay? Now, hindi po sa lahat ng pagkakataon perfect. Okay, na-attain ng isang secondary structure yung basic nila na features or yung basic nila na characteristics, okay, based on what we have described earlier. Okay? So may mga posibilidad na nagkakaroon ng irregularities, no? Irregularities, okay? Na sa mismong secondary structures. I repeat, not all the time yung mga characteristics ng alpha helix, beta pleated sheet na-attain, na-achieved. Okay? Irregularities may arise. Okay? Kumbaga hindi nasunod yung basic na description. Okay? So halimbawa, we have the most common. Most common ni siya, first two bullets. Yung first two bullets, common ni siya nga irregularities among alpha helices. Okay? Alpha helix. The most common one is 3:10 irregularities. Okay? Yung ibig sabihin ng 3:10, okay, 3:10 alpha helix, no? 3:10 helix structure. Okay? Yung 3 po dyan, these represent the the number of residue per turn. The number of residue per turn. Yan yung representation ng base 3 dyan. Okay? Yung 3, it represents residue per turn. Okay? Yung amino acid, number of residue per turn. Yung yung nasa baba naman, yung 10 dyan, it represents naman the number of residue, okay? Residue interval, okay? For the formation of hydrogen bond. I repeat, yung 10 dyan, it represents the number of residue interval, amino acid residue interval, bago yung formation ng hydrogen bond. Oh. So with that being said, regular or irregular ba yan sa isang helix structure? Regular or irregular ba yan sa isang helix structure? Obviously, tama. It is an irregularity. No? Irregularity yan ng isang helix structure. Ano ba dapat na basic turn? No? Number of turns sa isang alpha helix. Ano ba yung basic? Tama. Are you sure it's 4? Turn. Naglibog na mi ma'am. Tama, it's 3.6. Diba? 3.6. Yung turn ha, turns. We're talking about the turns. Okay? So, nabubuo yung turns, okay, with 3.6 residues. Okay, 3.6 amino acid residues. And what about for hydrogen bond? Ilan dapat yung amino acid residue? Okay, amino acid residue. Okay, bago mabuo yung hydrogen bond. Yes, 4. Yan yung basic, no? So, yun na nga, hindi naman all the time perfect, 'di ba? Hindi all the time. Meron ding sometimes hindi yan na susunod, no? Hindi yan na susunod. So, yan yung common, 3:10, no? Helix structure. I repeat ha, mga irregularities to, irregularities. So, the other irregularity sa alpha helix is 2:7, 4.4:16. So, you know that already. Ano ibig sabihin ng 2:7? I-apply nyo lang to. No? I-apply nyo lang yan. Okay, ano ibig sabihin ng 2:7, 4.4:16. So, you can rationalize that. Ano ibig sabihin yan. Okay? Then, for beta pleated sheet naman, ito yung common. Okay, beta. Okay, beta sheets. Common irregularity is yung beta bulge. Beta bulge. Okay? So, overall, we are done, no? With the secondary structures. Secondary structures. The alpha, beta helices. Okay? So, bago natin i-next slide, okay? Recall muna natin, no? Nasa secondary, sa buong secondary structures, okay? Na-discuss natin yung types, alpha and beta. Ano pa? Na-discuss natin yung loops, turns, bends. Tama? And yung irregularities. Okay? Yung irregularities. Now, supposedly, yung next na level, di ba dapat next level is tertiary. Diba? Tertiary structure, yung next level. However, meron po tayong midway. Okay? Meron pong arising, kumbaga, midway interface, okay? In between secondary and tertiary structure. Merong midway interface. Ang tawag natin dyan is super secondary structure. Okay, yung interface in between a secondary structure and a tertiary structure. So, ang tawag dyan, super secondary structure. So, I repeat, midway siya. Okay, midway. Okay. Um, yung super secondary structure, this is also known as motifs. Okay, this is also known as motifs, aka motifs. Now, under this, there are actually 4 common examples of super secondary structures. Okay? May 4 na examples. So, discuss natin siya. So, letter A, you have there the beta, okay, beta, alpha, beta. Diba? Beta, alpha, beta. Okay? So, bakit siya tinawag na beta, alpha, beta? Oh, look at the structure. Okay? You have a beta, 2 betas, okay? In between an alpha, no? Alpha helix. Oh, question. Yung beta structure dyan, ano yung direction? Parallel or anti-parallel? Parallel or anti-parallel? Very good. It's parallel. Kasi nga, same direction. Same direction. So, kaya siya tinawag na beta, alpha, beta. Based on the structure, ha? Based on the structure itself. Okay, so super secondary structure siya. Oh, for letter B, another example of super secondary structure, you have the beta hairpin. Bakit siya tinawag na beta hairpin? Yung mga biochemists, malawak yung imagination nila. So when they check the structure of a beta hairpin, it looks like a hairpin daw po. But me, personally, hindi ko alam kung may hairpin ba talaga na ganito. Ewan ko lang sa iba, di man gid ko hilig pudog hairpin. Ang akong gakakita every time magtanaw anang beta hairpin nga structure, mara siya paper clip. Yes, very good. Para sa ako, mara siya paper clip, no? Ayan. So, it's actually beta hairpin daw po. Okay, beta hairpin. Yan naman yung structure niya. Question. Sa beta hairpin, meron ba kayong nakikitang alpha helix sa structure? Yes or no? Alpha structure. Wala. Okay, wala. Puro beta. Okay, beta sheet. So, ayan. So, very good. So, somehow, you know already how to distinguish a beta sheet and versus to that of the alpha sheet. Okay. Next, the third one is alpha, alpha. So, obviously, dito naman, wala tayong makikitang beta sheets. Puro alpha to alpha. Alpha to alpha helix. So, ito yung sinasabi ko sa inyo. Yung nagko-connect sa kanila, di ba? Importante yung loops, turns, tsaka bends. Okay? Kasi nga, hindi magko-connect yung mga secondary structures kung wala yun. Okay? So, kumbaga, connection sila, bridges sila. And last, the fourth, okay, example of the supersecondary structure is the Greek key motif. Greek key motif. So in terms of the structure, kung makikita natin, di ba, alternating yung anti-parallel na strands, no? Puro beta, no? Puro beta sheets, anti-parallel. Anti-parallel. Okay? Now, ano yung connection ng structure na to sa name niya? Di ba yung name niya daw is Greek key motif. Okay, Greek key. Yung Greek key nga term, Greek key, historical yung term niyan ha. Okay? Sobrang wide ulit ng imagination ng mga biochemists upon checking the structure of this super secondary structure. Ilaha siyang na-compare dayon sa Greek key art. Okay? Sa isang Greek art. Yung Greek art. Okay? So, naamanggi Greek art, the Greek key. Okay? Wherein, ito yung isang Greek art, no? Ayan. Di manggid hilig ng mga art. Okay? So, for the chemist, ilang na-relate ba ang structure, no? Ang feature ng mismong strand na yan to the Greek key art. Okay, Greek key art. So, yan yung Greek key art. Okay? So, kung mas curious pagid mo, you can search more, no? Bakit, ano ba yung Greek key art? Wala na po akong more information ano yung nasa Greek key. Basta ang reason lang bakit tinawag siyang Greek key motif is look alike siya ng Greek art na yan. Okay? Greek art na yan. Okay? Ayan. So, yan lang po yung reason bakit tinawag siyang Greek key motif. Okay? Ayan. So, those are the four examples ng supersecondary structure. Okay, you have the beta alpha beta, beta hairpin, alpha alpha, and the Greek key motif. Okay, so overall done with the secondary structures. Okay, so let us proceed to the tertiary. Okay, tertiary structure tayo. Sa tertiary structure naman, this is actually a higher level already ng secondary structure. Kasi 'di ba, sa level of protein structure, level up man ta, pa-upgrade ng pa-upgrade. So pag tertiary na, higher level na siya ng tertiary structure. And sabi natin, 'di ba, yung key points sa tertiary structure, this is already a three-dimensional shape, no? 3D, three-dimensional shape of the protein. In fact, yung mga proteins in a tertiary structure, they are already said to be native. No, they are already native. Question, kapag native, functional or non-functional? Kapag native protein, functional na ba yan or non-functional? Wala, nakalimot na mi ma'am. Wala ginagtubag. Very good. Functional. Na, di ba ni sa atong introduction? Okay, so functional. Na? Kapag native, functional. Ano yung non-functional? Ano yung term kapag non-functional pa yung protein? It is said to be, it also starts with letter N, nascent. Very good, it's nascent. So yun lang yun. So pag tertiary structure ha, kapag nakaabot na ng tertiary structure, yung isang protein, most probably, majority sa kanila is already functional. Okay, so tertiary structure, the assembly, okay, of secondary structural units composed of matured na yung polypeptide chains and composed of domains already, domains. Okay, now I have mentioned earlier na tertiary structures, they are already functional. They are already said to be functional proteins. Okay, now since they are functionals, we can observe somehow certain na mga forces of attraction, mga bonds, no, nga pwedeng mabuo sa isang tertiary structure ng protein. So, aside atong na-mention nga H bonds, di ba, atong mga na-mention sa low level ng mga conformations ng protein, kay palagi na lang H bonds, may amide bond for the peptide bond, di ba? On top of that, pagdating ng tertiary structures, meron na tayong mga observable na different forces of attraction. Okay? So halimbawa, we could observe covalent bonding. Okay, we could observe covalent bonding. Okay, yung covalent bonding, this could be in a form of disulfide bonds. No? Disulfide bonds, pwede yan nabuo in between cysteine molecules. O question, ano yung atom present sa isang cysteine? Why is it capable siya mag-form ng disulfide bridges? Meron siyang cysteine has sulfur. Very good. Okay? Cysteine po, nga amino acid, cysteine molecules, meron siyang sulfur. Isa siyang sulfur-containing amino acid. That is why capable siya mag-form ng disulfide bridges. Okay, another Example ng forces of attraction that we usually observed sa tertiary structure is yung hydrogen bonding. O sa hydrogen bonding naman, aside atong hydrogen bond na bumubuo sa isang ordinary nga peptide chain, ang hydrogen bond may also form in between polar groups. Okay? Nabubuo siya in between polar groups of a side chain. So you know already what are examples of amino acids with the polar groups. So we have already discussed that no sa previous discussion natin. Then another force of attraction is an electrostatic attraction. So kapag electrostatic attraction, synonymous lang yan sa ionic ha. Ionic attraction. Kumbaga meron kang attraction between different ions, positive and negative ions. So kumbaga meron kang pole nga positive pole, negative pole. Okay? So most probably, you have there a protonated amino group. Kung protonated amino group, usually mga basic amino acid compose this, di ba? Meron ang mga basic amino acid ng positive portion sa ilahang side chain, tama? And then for the carboxyl group, okay? Carboxyl group sa side chain, makikita naman yan usually sa mga acidic, no? Acidic amino acid. So with that, electrostatic attraction may be formed. And take note, since polar siya, under din to sa polar, no? O, tingnan nyo yung previous notes nyo. Kapag polar side chain, pwede yan acidic or basic. So, all in all, polar to sila. Polar nga mga groups. Okay? Parehas lang yan, polar groups din yan. Okay? Now, with regards to this electrostatic attraction, this is actually occurs within the molecule of the protein. And I think, mentioned this sa one of the slides previously nga, sabi ko kapag polar side chain, polar amino acid, usually they have this, they are found within the molecule, no? Within the molecule of the protein. Okay? Not within the surface pala. Sorry. Okay, correction, surface of the protein. Not the interior, ha? Kasi pag within, interior. So surface meaning nasa outside ng protein. Kapag polar, di ba? Polar, outside, on the surface of the molecule. O kapag naman hydrophobic interactions, ito yung bumubuo ng non-polar. Hydrophobic, that's equivalent to non-polar residues. Okay, diba? Pag non-polar amino acid naman, non-polar side chains, they are found naman on the interior. Okay, na-discuss ko to. Question, na-discuss ko ba to sa last topic? Yes or no? Or nalimtan na? Kapag non-polar residue, they are found within the protein molecules. Okay, very good. So, very good, naaalala niyo pa, no? Okay. So, yan. So, don't forget this. Lumabas nga to sa isang quiz, di ba? Murag nigawas na siya sa quiz. Hydrophobic interaction. Hydrophobic non-polar residue. Interior. It is found in the interior portion of the protein. Okay. Sige. So, these are again examples of the forces of attraction that we usually observe in a tertiary structure. So, ito lang din yun. Okay, you have the hydrophobic interaction again among non-polar groups. Okay, then you have there the ionic bonds between charge na mga groups. Charge groups, so you have a protonated amino group, a deprotonated carboxyl group. Then you have here the disulfide bridge. Okay, so ito yung covalent bond. Halimbawa ng covalent bond. Alright, and then you have the hydrogen bonds. This occurs among polar groups. Okay, and yun na nga, since ang mga tertiary structures, they are already functional, diba? They are already functional, so meaning, meron na silang role in our body. No? Meron na silang role in our body. So specifically, for bullet 1, meron tayong mga proteins, no? Nga merong tertiary structures that already acts as enzymes, no? Okay, and we all know that enzymes, these are important, no, in our body. This is very important for catalytic reaction, no? Very important yung enzymes for catalytic reaction. And we also have proteins, okay, useful as receptors. Okay, so pag receptors, important yan for binding. Important yun for binding of ligands. Binding of ligands. Another function of tertiary proteins, okay, with a tertiary structure na proteins is they're able to anchor, okay, anchor to a, they could anchor a protein, no? Meaning, ani, transporter ba? Transporter sila. Okay? Transporter. Then, lastly, they are used as regulators, no? Regulators. So, mga regulatory protein naman yung pasok dito. So, halimbawa, yung nasa slides, these are enzymes, no? Ending in A's, these are enzymes. So, these are already functional, no? These are functionals. Okay, so tertiary structure yung pinapresent nila dyan. Okay? Now, yung proteins, okay, with three-dimensional structure, they actually fall into two main types. Okay? So, with regards to this slide, meron tayong thorough discussion on this, ha? Nakasulat man po sa task sheet number 4, di ba? Next meeting, discuss natin yung globular tsaka fibrous proteins. So for now, you can just read this, okay? Kasi may thorough discussion din naman tayo on this specific concept sa globular and fibrous proteins. So just an information lang na yung mga tertiary structures, they are very much well observed on globular and fibrous proteins. Okay? Kasi marami tayong globular proteins like hemoglobin, no? Nga functional, no? Hemoglobin is very important sa ato ang blood, no? Sa atong red blood cells as oxygen carrier. Fibrous proteins, meron tayong halimbawa ng mga fibrous proteins, very important also in our body. Functional na kumbaga. Okay? Then lastly, last na po tayo sa quaternary structures naman. Okay, for quaternary structures, we have, ano naman yung kaibahan niya sa tertiary? Sa quaternary structure po, okay, ganun pa din naman, 3D, same lang 3D, representation, okay, 3D, native na din, native, functional na kumbaga ba, okay, functional na yung protein, kasi nga, highest level na ba to, di ba? Highest level na po yung quaternary structure ng isang protein. Final na, bali, final level. So, sobrang functional na yan ng protein. Okay? Now, ang question ngayon, ano yung pinagkaiba nila ngayon ng tertiary structure? Okay. Take note, sa mga klase ng proteins po, meron tayong mga certain proteins. I repeat, meron tayong mga certain proteins kung saan na kahit isang polypeptide chain lang meron sila, kahit isang polypeptide chain lang meron sila, they are already functional. Okay? Now, if that is the case, that is tertiary structure. Nakuha? I repeat, kapag tertiary structure, okay, halimbawa nito is yung mga proteins na isa lang yung polypeptide chain niya, pero already na-attain na yan yung functional state. Native na siya kumbaga. Okay, kahit isa lang yung polypeptide chain niya. Now, in that case, kung na-attain na niya gani, no, ang functional state, then isa ra iyahang polypeptide chain, iyang highest level ana is only tertiary. A tertiary structure. Okay, mo na siya iyang highest state. Kapag isa lang yung polypeptide chain, attain na niya yung functional state. Native na siya, na-mature na siya ba? Okay, at tertiary state. So that's for tertiary structure. Now, for quaternary structure naman, this time it consists of more than one polypeptide chain. I repeat, quaternary structure po, this time more than one polypeptide chain. Kaya nga di ba yung key points natin sa quaternary structure, balikan niyo yung overview natin sa 4 na levels of structure. Sabi natin na ang quaternary structure, 3D na rin siya, but this time more than one. It consists of more than one polypeptide chain. Okay? And this is, yes, functional na native. Okay? Kung matured na kumbaga yung polypeptide chain. And with regards to that sets of polypeptide chain in a quaternary structure, ang tawag po natin sa each polypeptide chain dyan is subunit. I repeat, sa isang quaternary structure, halimbawa, 'di ba, meron kang isa, dalawa, tatlo, apat, no? Apat na polypeptide chain, okay? Then native yung protein, so quaternary na to, quaternary structure. Ang tawag natin sa per polypeptide chain dyan is subunit. Okay? Ang tawag natin dyan is subunit. O, ano naman yung tawag natin sa each amino acid in a peptide chain? Ano yung tawag natin sa each amino acid sa each, tama, sa polypeptide chain? Residue or ano yung other term? Residue or moiety. Moiety. Okay? Ayan. Sige. So ano yung halimbawa? Pinaka-common na halimbawa po ng quaternary structure is yung hemoglobin. This is the most common yun, no? Na example ng quaternary structure, hemoglobin. We will discuss na at a segment nga ma-discuss na to all about yun, this examples of protein. Under na siya sa globular protein, ang hemoglobin. So for now, just know that this is a quaternary structure. So kung yung tag-quaternary structure si hemoglobin, so marami siyang polypeptide chain and functional na siya. Native, matured na yung mga polypeptide chain. That illustration is actually the hemoglobin. Okay? Hemoglobin po yan. Okay? So, as mentioned earlier, ang quaternary structures, they are already consist of multiple polypeptide chains. Therefore, we can identify them. We can classify also quaternary structure based on what it composes. If it composed of identical ba lahat ng mga subunits or different ba yung mga subunits. So, kapag sinabing sa isang quaternary structure, yung mga polypeptide chains, yung mga subunits identical, then that's homo. This is somehow a common sense, diba? Homo means uniform. Homo means uniform, the same. Okay, uniform, the same. Okay, so homo oligomers. Okay, homo oligomers. Then, kapag naman ang isang protein containing several different. Distinct means different, no? Different, magkakaiba yung polypeptide chains nila. Hetero naman yan. Hetero naman yung polypeptide chain. Yung ibig sabihin dito na polypeptide chain, yung halimbawa kapag identical, puro alpha. Okay, so kapag ganyan, alpha, 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 ganun, then that is identical. Paano naman kapag alpha, then may beta, okay, then another beta. So kapag ganyan, yung mga polypeptide chain dyan, nagkakaiba yung structure niya, then that is hetero-oligomers already. Okay, hetero-oligomers already. Okay? Sige. So ito yung sabi ko kanina, hemoglobin is the most common protein with a quaternary structure. So hemoglobin is actually the oxygen carrier protein ng blood. Okay? Kaya po kulay pula, no? Kaya po kulay pula yung blood. That is actually because of hemoglobin. Okay? So ano yung structure ng ano ba yung meron, no, sa hemoglobin? Kumusta yung polypeptide chain niya? This is actually has the subunits of 2 alpha and 2 beta. Okay, I repeat, 2 alpha, 2 beta subunits. Yan yung meron si hemoglobin. Okay? Mag-discuss kita ani ha. Ayaw mo ka problema dira. Mag-discuss sa next meeting. Okay? So, with that being said, kung you have 2 alpha, 2 beta, anong klaseng oligomer ito? Hetero-homo protein. Hetero or homo oligomer? Very good. This is hetero. Hetero-oligomer. Hetero-oligomer. Okay. Kapag hemoglobin po In some other references, they also call this as tetramer, no? Tetrameric. Okay, tetramer, tetrameric protein. It is already understandable bakit tetra, di ba? Bakit tetra? Kasi meron siyang apat na subunits, no? In a form of alpha, dalawa. In a form of beta, dalawa. So, apat lahat yan. Okay?